In this video, we are going to talk about the biochemistry of carbohydrates. The other name for carbohydrates is saccharides, and they are composed of carbon, hydrogen, and oxygen. And the general formula of carbohydrates is CH2O by N, and the N can be 3 or 4 or more. Carbohydrates have several functions. For example, they are a source of calories, they store energy, and they form several things, such as the fibrous cellulose of plants, and the cellulose is one of the carbohydrates, and we are going to talk about the structure of cellulose later in that video. They also form the exoskeleton of insects, and they form the cell wall of bacteria. The carbohydrates are classified into monosaccharides and disaccharides and polysaccharides. The monosaccharides, they are the simple units. They are the simplest units of the carbohydrates. And the disaccharides, di means two, so they are composed of 2 units of monosaccharides. The polysaccharides, they are composed of more than 10 monosaccharides units. So the monosaccharides, they are the building blocks of the carbohydrates. Let's start with the monosaccharides. So the monosaccharides, they are classified according to the number of carbon atoms forming it. So, for example, if you have a 3-carbon monosaccharide, the generic name for that would be triose, and an example for that would be glyceraldehyde. And if you have a 5-carbon monosaccharide, the generic name for that would be pentose, and an example would be ribose. A 6-carbon monosaccharide will be hexose, and an example for hexose is glucose. The monosaccharides can be found in 2 forms. The first one is the linear form, and the second one is the cyclic form. Both of these monosaccharides are composed of 6 carbon atoms, and they are both glucose, but they are found in different forms. So to turn a linear form into a cyclic form, we would do this reaction. So there is a reaction between the first carbon and the OH group of the 5th carbon. Right here you can see the first carbon with the 5th carbon. There is OH of the 5th carbon, and there is a reaction between the first carbon and the OH group of the 5th carbon atom. So the first carbon here, it is called the anomeric carbon atom. Monosaccharide isomers, they are different compounds, but they have the same molecular formula. So how is that possible? Let's see here. The first type of monosaccharide isomers is epimers, and before we start with the definition, let's take a look here. So we have glucose and galactose. Both of glucose and galactose have the same number of carbon atoms, which is 6, and the same number of oxygen atoms, and the same number of hydrogen atoms, which means that they have the same molecular formula. We said that they are different compounds, so how are they different? If you look at the 4th carbon and look at the hydroxyl group attached to it, you would see that it's on the right of glucose and on the left of galactose. So there is difference in the configuration around one carbon, which is the 4th carbon right here. But that carbon can't be the anomeric carbon, which is the first carbon in this case. Another type of isomers would be aldose ketose isomers, and they differ in the functional group. So it can be aldehyde group, such as in glucose, or ketone group, such as in fructose. So the first carbon here with oxygen and hydrogen, this is considered an aldehyde group, and the second carbon here with the oxygen is considered a ketone group. So this is the aldehyde group, and the anomeric carbon in that case will be the first carbon, and this is the ketone group, and the anomeric carbon here, it is the second carbon, not the first carbon. We also have the alpha and beta isomers, and they only differ in the orientation of the hydroxyl group attached to the anomeric carbon. So if this hydroxyl group is in the opposite direction of the last hydroxyl group, this is considered alpha, and if it's in the same direction, this is considered a beta glucose. Now let's get to the disaccharide, and as we said before, the disaccharides, they have 2 monosaccharides, and they are linked together by a bond, and this bond is called a glycosidic bond. The first disaccharide we have here is the maltose, and the maltose is formed of 2 glucose, specifically alpha glucose. And there is a reaction between the hydroxyl group of the first carbon and the hydroxyl group of the 4th carbon of the other glucose molecule. And there we have it. We have the maltose, and it has a bond here. It is alpha 1,2,4 glycosidic bond, because it's a bond between the first carbon of the first alpha glucose and the 4th carbon of the second alpha glucose. So it is alpha, both alpha 1,2,4 glycosidic bond. The second disaccharide we have here is the sucrose, and the sucrose is formed of alpha glucose and beta fructose. And the reaction here is between the hydroxyl group of the first carbon of the alpha glucose and the hydroxyl group of the second carbon of the beta fructose. And that gives us sucrose. The bond in sucrose, it is between the hydroxyl group of the first carbon of alpha, so it is alpha 1, and the hydroxyl group of the second carbon of beta, so it is beta 2. So it is alpha 1, beta 2 glycosidic bond or glycosidic linkage. And we have another disaccharide, which is lactose, and the lactose is formed of beta galactose and beta glucose. And the reaction here is between the hydroxyl group of the first carbon of beta galactose and the hydroxyl group of the fourth carbon of the beta glucose. So the bond here is beta 1,2,4 glycosidic bond. Now let's get to the polysaccharides, and they are classified into homopolysaccharides and heteropolysaccharides. So homo means the same and hetero means different. So here it contains only one type of monosaccharide units, but here it's formed of different types or more than one type of monosaccharide units. Both of homopolysaccharides and heteropolysaccharides, they can be branched or unbranched. So this is branched and this is unbranched homopolysaccharide and heteropolysaccharide. We will talk about the heteropolysaccharides in another video, so let's talk about the homopolysaccharides first. So the homopolysaccharides can be different according to the monosaccharide that they have in their structure. For example, glucosanes, they are a group of homopolysaccharides. They contain only glucose units in their structure. We have starch, which is a storage of carbohydrates in plants, and it is formed only of glucose units. Also glycogen, it is formed of glucose units and it stores the carbohydrates in animals. And we have cellulose, which enters in the structure of the plant cell walls. Another group would be fructosanes, which contain only fructose units. And for example, we have inulin. It has only fructose units in its structure. So glucosanes, the first one is starch. And the starch is formed of alpha glucose units and it's linked by alpha 1,2,4 glycosidic bonds. So it is arranged into two structures. The first one is the amylose and the second one is the amylopectin. The amylose is unbranched and the amylopectin is branched. And the amylose makes 20 to 30% of the starch structure, and the amylopectin makes 70 to 80% of the starch structure. So the bond here is alpha 1,2,4 glycosidic bond, but the bond at the branching point is alpha 1,2,6 glycosidic bond, as you can see here. Next we have the glycogen, and the glycogen, its structure is similar to the amylopectin, which means it is branched and it is highly branched. That's the only difference here, that it's highly branched. So as you can see here, there is a branch every 7 to 11 glucose units. As you can see here, there is 7 to 11 glucose units, and you have a branch here and a branch here. So there is a branch every 7 to 11 glucose units. But in the amylopectin, there is about a branch every 20 to 30 glucose units. Lastly, we have the cellulose, and the cellulose is linear, which means that it's unbranched. As you can see here, there is no branch in the structure of cellulose. And the bond here is beta 1,2,4 glycosidic bond, which is different from alpha, as you can see here.